My name is Matthew Albrecht, and I'm a plant ecologist, conservation biologist at the Missouri Botanical Garden in St. Louis, Missouri. And what we're doing is we're working on a long-term research project with an endangered plant species, Pyne's ground plum. This is a species that is globally endangered. It's only found here in the Tennessee Central Basin. It's naturally known from about a half a dozen sites. And part of our research is trying to understand how to recover this species in the wild. And so what we're doing is we know historically from our barium specimens that this species once grew more widely and broadly within this Tennessee Central Basin range. And because of habitat loss and habitat degradation, many of those sites have been lost over time, and so the species has been extirpated from those locations. And so what we're trying to do is reestablish it within its historic geographic range. And so this is one of our sites that we began reintroducing the species to. This is a natural area managed by the Tennessee Department of Environment and Conservation. And we started this project in 2012, and we have about 125 to 130 plants here that we've outplanted over multiple years, 2012 and 2016, and every year we come out and we have tagged plants here. You can see these blue tags that have a unique identifier on them, and for each one of these tagged plants we come out to determine whether it's survived, whether it's flowering, and then whether those flowers go on to produce fruits. And so we're going to come back out here in May to look at whether or not these were pollinated and they produce fruits. And so over time, we can then begin to learn how the biology of the species and how the species behaves across these different sites. And we've been kind of conducting these as long-term research experiments. And so if you would have come out here last year, about half of these plants would have been caged to keep herbivores from consuming the plants. Herbivores are a major sort of threat to this species because it emerges so early in spring, and the herbivores like to munch the new growth on plants, which basically sets them back and prevents them from flowering. And so this particular example is a group of plants that was probably caged for the past 4 years, and so you have really good flowering this year even though the cage is off of it, because the plants have been growing large over time, and they have enough resources now to flower. And so our goal is to see whether or not these plants can survive without their cages.
So that's kind of the immediate experiment, so to speak.
Yes, correct. We've also been researching with, so half of these plants were inoculated with soil rhizobia. So this is a legume, and it forms a symbiotic relationship with rhizobia bacteria below ground. And so we've been looking at how these soil microbes may be influencing the species' growth and survival. And so we've been tracking, we know whether or not which plants were inoculated with rhizobia and which ones were not, and that will help us learn whether or not we should be inoculating them during our reintroduction program or not.
Fantastic. So you had a control set and an experimental set.
Exactly. So about half the plants were inoculated, the other half were not, and then in each one of those groups, about half of those plants were caged and the other half were not caged. So it's a 2x2 experiment. So we're manipulating whether or not herbivores have access to plants and whether or not plants have rhizobia or not.
So a two-variable experiment.
Exactly.
And I guess for these kinds of situations where you're not right there, you have a limit to resources and time, you sometimes kind of have to do 2 and 3 variable things just to get data in and get your trials in. And
yeah, and we've repeated this across multiple sites. And so each time we're doing new reintroductions, we're sort of taking the information we learned from these older ones and from the experiments and applying them to our new reintroductions and trying to gain greater success over time as we go through the program.
Do you have any sort of, I guess you could just say, provisional trends that you're seeing so far, although we know we're not really done with this experiment by any means?
So in terms of caging is where we see the biggest effect. And so plants that have been caged perform much better than those plants that haven't been caged. So they tend to have greater growth and greater reproductive output, flowering and fruiting.
Go ahead.
No, yeah, and the rhizobia is a little bit more mixed. We're not seeing quite as strong of effect as we're seeing with caging.
Yeah, so far that's a perhaps modest effect.
Exactly.
At that time. So how long do you expect this to keep going, or did y'all have a rough end time for this planned, or?
Well, we're funded right now until 2023, and we're working with the U.S. Fish and Wildlife Service and again the Tennessee Department of Environment and Conservation on this project. The goal is to ultimately meet the recovery criteria on the recovery plan laid out by the federal government for the species, which I think X number of populations need to be reestablished within its historic range. So our goal is to try to meet that criteria down the road. So we'll be working on this for many more years.
So this is part of the implementation of a Endangered Species Act recovery plan.
Exactly.
Excellent. And so in these kind of in these plans, there's sort of like you sort of mentioned, like there's something of a benchmark for success, and you're looking to try to reach that that benchmark, so to speak. And then in theory, if the benchmark is met, it might get taken off the list and be considered a recovery success.
Exactly.
Fantastic.